Hi, this tutorial is designed for individuals who are non-experts in metabolomics who might be analyzing metabolomics datasets. The goal of this presentation is to help you better understand the untargeted metabolomics data that you may be analyzing. This is an example of a dataset that you may receive. This is what is called a fully untargeted metabolomics dataset. You'll see it's generally going to be a very large spreadsheet. In the first two columns, you'll see information such as the M/Z and the retention time. And then you may see other columns that contain very large numbers. And on the spreadsheet, you may or may not see metabolite names associated with each of the rows. So for somebody who's an expert in metabolomics, this all might make a lot of sense. But for somebody who's not an expert, there may be many questions that you might have about how to properly analyze the data that you're receiving. For example, the spreadsheet is often called a feature table. So you may be asking, what exactly is a feature? Those two columns, the M/Z and retention time, what do they actually reflect? Does every feature, or you may also hear this referred to as a peak? Do each of these features and peaks reflect metabolites? And then you may have received separate spreadsheets for different, quote, columns and modes. So you may be wondering what this might mean. And then finally, I'll explain how we take the features or the peaks and we identify metabolites from these. To answer these questions, it's important to understand in a very basic sense how LC-MS, untargeted metabolomics, is accomplished in the lab. For whatever sample you're analyzing, it first goes through a series of preparation steps, and then it gets injected into the LC-MS. The first important step is that your metabolites need to be separated out in some way, and this is achieved through what is called chromatography. Two very common approaches are known as the hydrophilic interaction liquid chromatography, or HELIC, or the reverse phase chromatography approach. Very simplistically, how this works, for example, for HELIC, is that it separates out the metabolites in your sample based upon the chemical properties of the metabolites. So for example, the HELIC column is what is known as a hydrophilic column. What this means is that the metabolites in your sample that are hydrophilic really want to associate with the column. So what happens is that you put your metabolites into the sample, or into the sample column, and then what you do is you inject a solvent that's hydrophobic, meaning that all of the metabolites in your sample that are in the column that are hydrophobic want to associate with the solvent, whereas the hydrophilic metabolites, they're going to be interacting with the column more strongly. What this means is that the hydrophobic metabolites tend to be eluted from the column first, whereas the hydrophilic metabolites tend to hang out in the column for longer, meaning that the retention time, which is one of those columns in your spreadsheet, it just reflects how long the metabolite hangs out in the column, which is a reflection, at least in the HELIC column, as to how hydrophobic or hydrophilic it is. You may wonder why we need different types of columns. And the reasoning for this is that certain columns can better detect and characterize various different types of metabolites. So if you have two different chromatography approaches, it can really capture all of the metabolites that are in your sample. The next important step in this process is what is known as electrospray ionization. It's important to note that metabolites that aren't charged can't be detected by the mass spectrometer. So what electrospray ionization, or ESI, does is it generates charged ions from your metabolites. For example, take a metabolite with a mass of 146.069. This metabolite plus an isotope of this metabolite may be present in your sample. These charged metabolites are not what you're going to be detecting. Instead, through electrospray ionization, what happens is that these metabolites get converted to these charged ions or adducts. So for example, you're not going to be detecting your metabolite, but you'll be detecting is your metabolite plus a hydrogen or plus a sodium. And these are all charged. You'll see that the M over Z, which reflects the mass to charge, reflects the mass of your metabolite plus the mass of each of these adducts that are attached to these metabolites. So all of these adducts and fragments and potentially isotopes are what you're actually detecting. These parent metabolites that are not charged are not what you're measuring. So take, for example, the metabolite cholesterol-phosphocholine, which has a mass of 257.1028. You're not going to be detecting a signal, which may be referred to as a feature or a peak, that corresponds to 257.1028. What you'll instead be detecting is a series of peaks or features that reflect the different adducts that are formed after ESI, plus possibly some of the isotopes and fragments that are present as well. In this example, cholesterol-phosphocholine is represented in the data set not by one peak, but by 5 peaks that correspond to 3 different adducts that are formed, plus the isotopes of these. What's complicated is that when you have an LC-MS untargeted metabolomics dataset, what you're getting is just a jumbled mixture of all of the adducts and fragments and isotopes that are present from all of the metabolites that are in your sample. You may or may not have information as to which of these peaks or features correspond to which metabolites. So generally what you get is information on the peak or the feature, which are all defined by unique M over Z and retention times. So you know that there's definitely a peak there that has a particular mass to charge and retention time, but you may or may not know what the metabolite is. So just to reiterate, these peaks or features or signals or whatever you may call them, they reflect metabolites, and they're defined by 3 characteristics. You have your M over Z, or your mass over charge, which is what's detected by the mass spectrometer, or the MS. Their retention time, which is a reflection of the chemical properties that are used for the liquid chromatography separation. And then third is the intensity, which is just the abundance of that particular ion in your sample. So if we go back to the spreadsheet that I showed you at first, now you actually know what all of these things mean. So what you can see is that each of the rows of the spreadsheet reflect a peak or a feature. They're defined by the M over Z, or the mass over charge, and the retention time, which are unique paired characteristics for each of these features. And then the abundances, or the amount of that particular ion that's present in each of the samples, is in these subsequent columns. After the data is generated, the data goes through a series of pre-processing steps before it arrives in your hands. As the person analyzing the data, it's really important to understand what exactly the lab did in these pre-processing steps before the data got into your hands. Very simplistically, the first step is that the raw data first goes through what is called the alignment and detection step. This is generally achieved through a package called XCMS. And what happens is that it takes the chromatograms and it aligns all of the peaks across all of the samples, and it is able to say whether a peak is actually a peak, and then it detects the abundances of each of these peaks in all of the samples. Next, generally there's a step for batch correction. In the LC-MS workflow, there can be quite a bit of analytical variation in the intensities that are detected between batches. So generally, what the lab will do is they will do some sort of a batch correction, like for example through combat, as a statistical approach to reduce the variation between batches. The success of this statistical batch correction is contingent upon the samples being randomized within the study. And finally, there are a series of imputation and normalization steps that are going to vary from lab to lab. So it is very, very important that you understand what was done in the lab. The first is there's usually some sort of imputation for what are called missing values. These are values where the peak in that sample may have been below the level that could be reliably detected. And then there may be some values that are missing because of technical issues. There are a variety of different approaches that could be used for imputation, such as leaving these values as NA or missing, imputing with a small value, or doing a more complicated approach such as k-nearest neighbors or a random forest algorithm. The next step is to take out what are called unimportant peaks. There are a lot of uninformative peaks that may not reflect metabolites or may be redundant. So there are a variety of different approaches to filter out some of these uninformative peaks, including taking out some of the peaks that have uniformly small values, some that have relatively high levels of missing values across the samples, or the peaks that have near constant variation or low repeatability across your samples. And finally, there are a number of different normalization approaches for sample-wise and metabolite-wise normalization to improve comparability, in addition to different transformations that may be applied if the data look like they're not normally distributed, which they probably will not be. So going back to the spreadsheet that we were presented with earlier, all of these pre-processing steps were used to generate the spreadsheet. So again, it is really important that you talk to the lab and you understand exactly what pre-processing steps went into the generation of your data. To review the important terms that we went over today, the first is what is known as the feature or the peak. This is what you get after processing the chromatogram. These are in the spreadsheet as the rows, which are defined by their M over Z or mass to charge, and the retention times in the column. They're detected or measured as ion abundances. As we went over, they don't match one-to-one with metabolites. So metabolites may be reflected by several different features or peaks that are different adducts and isotopes from the metabolites, in addition to there may be also like fragments in there and noise. The next important concept is what's known as the chromatography column. This is just a method for how the lab separates out your metabolites. Usually labs use two different types, usually a hydrophilic and a hydrophobic column. There are two types that are used because these columns can better capture different metabolite types. So by using both, you get a really broad coverage, but again, there's like overlap in the metabolites that are detected. And then finally, the mode, or the electrospray ionization mode, the positive or negative, it's how the lab makes the metabolites charged for detection. Again, this is through ESI, and it could be positive or negative. And the reason that we do both is generally because there are certain metabolites that may be better detected in one mode versus the other. The last thing you may be wondering is how labs actually report metabolite names. How do you actually get from the features or the peaks to metabolites? This will be covered in an upcoming tutorial where we'll talk about matching against analytical standard libraries, the process of going through MS, and then annotation by accurate mass.